The secret life of cancer cells, how autophagy drives their spread. Cancer is feared because of its ability to spread, invading new territories in the body. Imagine cancer cells as explorers, eager to break away from their original home and colonize distant lands. But how do these cells gain the energy and tools to undertake such perilous journeys? The answer lies in a remarkable survival process called autophagy. Autophagy, which literally means self-eating, is a cellular housekeeping process. Normally, it helps healthy cells recycle damaged parts, such as faulty mitochondria or stressed-out organelles, keeping them clean and functional. But cancer cells, always clever survivors, have hijacked autophagy, turning it into their own superpower to facilitate their dangerous migrations. Before tumor cells can spread, they must first transform, gaining mobility and a sense of direction. Think of a cancer cell as a tiny vehicle. First, it pushes forward its leading edge like a bulldozer blade. Then, special molecules called integrins latch onto surrounding tissue, forming temporary anchors known as focal adhesions. Inside the cell, a protein scaffold, the actin cytoskeleton, strengthens and expands, giving the cell shape and stability. With a powerful contraction, the cell propels itself forward. Finally, its trailing end detaches, allowing the cell to move on and repeat the cycle. Cancer cells have multiple modes of movement. Some crawl like amoebas, others stretch out like mesenchymal cells, and some use hybrid strategies. Yet regardless of the type, one thing remains constant. Autophagy is essential to their success. For years, researchers viewed autophagy mostly as a simple cleanup mechanism, until its crucial role in cancer progression earned the 2016 Nobel Prize. Later, scientists discovered another shocking twist. Autophagy is not just about survival. It actively helps cancer cells invade new territories. Autophagy helps cancer cells in two main ways. First, it provides energy and materials under stressful conditions. Imagine cancer cells growing rapidly and quickly running out of oxygen and nutrients. To survive this crisis, they use autophagy to recycle their internal components, like mitochondria or damaged organelles, generating energy needed for their migration. Second, autophagy influences how cancer cells interact with their surroundings. It regulates focal adhesions, those critical contact points that cells use to move forward. When autophagy is functioning normally, these focal adhesions are dynamic, constantly formed and broken down, enabling smooth movement. Blocking autophagy halts this cycle, leaving cancer cells stuck, unable to move forward. Another key player in cancer metastasis is the process known as epithelial mesenchymal transition, or EMT. During EMT, cells that were stationary and orderly, epithelial cells, transform into mobile, invasive cells, mesenchymal cells. Think of EMT as cells abandoning their quiet life in a structured community, embracing a nomadic existence. This transformation involves losing markers of their original identity, such as a protein called E-cadherin, and adopting new traits, including increased levels of N-cadherin, vimentin, and enzymes known as matrix metalloproteinases. Cancer cells rely on several signaling pathways, WNT, TGF-beta, and others, to make this shift happen. Interestingly, autophagy can both enhance and restrain EMT. One protein, P62, also called SCIC-STEM1, acts as an autophagy adapter that stabilizes twist, an essential factor driving EMT. But paradoxically, by stabilizing twist, autophagy-related mechanisms involving P62 can also suppress EMT, limiting cell migration. Thus, autophagy seems to play a complex dual role, sometimes promoting, sometimes restraining EMT depending on the context. At the heart of metastasis are cancer stem cells. Cancer stem cells behave like seeds that can travel to distant locations, planting new tumors and driving cancer's relentless progression. EMT supports cancer stem cells by activating transcription factors like SLUG, which boosts genes responsible for self-renewal, growth, and aggressive behavior. Autophagy further strengthens cancer stem cells, equipping them with tools for survival and invasion. By helping cancer cells respond swiftly to environmental stressors, autophagy ensures cancer stem cells remain potent, ready to conquer new territories. Given its crucial role in cancer's deadly journey, autophagy has become an attractive target for therapies. Studies have shown that blocking autophagy can prevent cells from migrating and reduce metastasis. For instance, inhibiting a protein called focal adhesion kinase activates another protein, SRC kinase, suppressing autophagy and stopping cancer cell movement. By understanding these connections, researchers hope to develop strategies to dismantle cancer's survival mechanisms, halting metastasis, and improving patient survival rates dramatically. In short, autophagy, a seemingly innocent recycling process, is exploited by cancer cells, providing them with energy and coordination needed to invade distant organs. As researchers unravel the complexities of autophagy, we move closer to discovering powerful new ways to stop cancer's deadly spread.